really something very exciting that we are going to start now. And before everything else, I would like to acknowledge as strongly as possible the contribution of Or for this event, Or Spiegel. So Or was the main driver. He initiated, he had the idea to come up with this school. He got some money from the university, and we added lots of funding from the Minerva Center funding, and then he organized the whole schedule and got involved with every single detail with this course. And so, in the name of everybody here, thank you very, very much for your wonderful work. And also, I would like to thank our superb speakers, Otso and Luca. Where's Luca? Good morning. And I think it is a wonderful opportunity for all of us to get into starting from basic analysis of movements, and then, as Luca said yesterday, there will be an acceleration towards some advanced methods. Not too advanced and not too mathematical, but I think you will all see the logic of these methods, and will do your best to follow the material. It should not be too difficult. I'm sure that all of you can follow and do the hands-on exercise. That's a very important part of this course, that not only you learn the theory or the ideas, but also practice with these tools. So this is the detailed schedule of the course, and you all seen that, right? And you know that by heart. Okay, so I will not get into the details, just to say that every day we start, in general, the day with some lectures in the morning, and then afternoon we'll have exercises. It's not every day like that, but that's a general scheme. And maybe one last thing I need to say in the introduction is that you are requested to ask questions. Okay? If you feel that it's going too much faster, or there are some critical points that you do not understand, raise your hands and ask the question. Okay? Most of the course will be delivered by Otso and by Luca. So they are looking for your questions. Okay? They will stop the run and address your questions. And so this is one point. And the second point is that, unfortunately, today everything is closed, as you know. So you will have to arrange yourself with some things that you brought with you, I hope. And then that's the plan for today. So what's that? Yeah. And of course, sorry, thank you for correcting me. And Henjo also is a teacher in this course. He will give the exercises, right? All exercises. And yeah. And thank you for coming too. And so we'll start, okay? This course is about the analysis of movement data, and the study of movement of animals and plants is not new. It has very strong roots in, I think, every attempt of mankind to understand nature. And because movement is one of the most fundamental and basic features of life. And it is defined as the change in the spatial position over time. And it characterizes every single organism, as far as I know, at least in one stage of its life cycle moves. Some quite sessile organisms, so-called, are mobile at the beginning of the life mostly, like seeds of plants and larvae of corals and so on. And we know that movement is critical for survival, for reproduction. It's a major determinant of fitness. And movement is a key process in evolution. Many of the main events in evolution have to do with a selection to facilitate movements, as this lineage of dinosaurs from which the modern birds evolved. You know, the shift from the sea to the land is much about adapting different kinds of movements. And we see that again and again and again when we consider macro and micro evolutionary processes. And of course, also in ecological systems, movement is of critical importance. And these are just some of the few examples in which movement is the key for processes that shape ecosystem functioning, community diversity, and so on and so forth. So with all that in mind, this is well recognized. It's known and has been studied intensively and deeply. And with all that in mind, it is also important to recognize that movement is critically important for human life. And we now face major problems, major environmental problems, and we can divide these major environmental problems to those that associated with movement and movement is insufficient, and those that associated with movement of organism and movement is excessive, it's too much. And for example, for the first type, global warming is shifting the climate regions, and we are concerned whether or not animals and plants will be able to track this shift. I will talk about that a little in more detail later on. So here we are concerned that movement might not be sufficient to follow the environmental changes. And this is another kind of global concern. We have habitat fragmentation and habitat loss that creates this patchiness, and now the concern is whether or not animals and plants will be able to move in this fragmented landscape and maintain metapopulation dynamics and so on. And the other type of concerns is that movement is so superb and so fantastic in some organisms that they create problems of health, spread of pathogens and pests and so on, and of invasive species. And all of these concern as major economic influences. So these are four of the major problems that we face in Earth today. And movement is the fundamental process, movement of organism is the fundamental process that drives these major concerns. So we have a strong motivation to study movement. It is a characteristic of every organism. It is important evolutionary and ecologically and also practically. So indeed, it has been studied in great detail, and these are just few of the many books on this subject. And Marcel Holyoke led this review of papers in the major journals of ecology and genetics and zoology and botany and all these, about 500 journals. And we were very, very surprised to see that this huge number of papers, 26,000 papers in 10 years on movement, and you can see that the absolute number is increasing from about 2,000 per year to 4,000 per year, and not only the absolute number, also the proportion is increasing from about 7% to 10%. So 10%, maybe now it's even more, 10% of all papers in the very general disciplines of ecology, environment, zoology, biology, genetics are directly or indirectly related to the movement of organisms. And with that, it is now the time to consider in general the common reason for moving with any movement, whatever. And this sounds like very modern and timely argument, but it was phrased by this person, Aristotle, 2,300 years ago. This is the opening sentence of this book on the motion of animals. And Aristotle studied the movement of animals, and he actually defined, not formally, the laws of some of the laws of motion of Newton, and then he said, okay, I learned enough. It is now the time to do the big integration, and this is very much like the philosophy of Aristotle. You know that he was arguing for the importance of basic principles. We need to find basic principles that drive the natural world. And with that approach, that was the main message that brought to the front stage of what can be called science at these days. However, the progress at this front was very, very limited, if at all, because science is moving in other directions. Science is moving towards more specialized things. And what we do and what we teach you to do, and what I think you will teach your students to do, is to get to be expert in a particular system, because you need to understand the system very well. So you need to know very well the component of the system, what drives the systems, and so on and so forth. But this trend in science, which is all over branches of science, people are doing science in this way, is of course very beneficial. We got wonderful insights, but on the other hand, it has cost, and the cost is that we're getting farther away from these general insight and integrations, and so there are, of course, attempts to develop new theory, very general theory, and so on, and the theory of natural selection is, of course, the leading theory that you can see. And when you think about natural selection and Darwin's contribution, you can appreciate the importance of developing a general theory. And so, speaking about Darwin, at the time of Darwin, this person, Robert Brown, was actually a colleague of Darwin. And you know the famous discovery of Robert Brown, that the grain of pollen of this plant seemed to be moving in what was conceived as steady medium. Without the movement of the water or the liquid that was there, and this is actually what what can be seen from the microscope that Robert Brandt was using. This picture was taken in the same microscope. And now we know that the pollen is moving because of the movement of the water molecules. But this was, what's that? That's always here? Okay. But this insight is not trivial. And that was the insight of this person, Albert Einstein. And for other reasons, not to study movement, but for other very important reasons, Einstein began a study of Brownian motion. And this paper in 1906, the marvelous year of Einstein, where he published all these 4 important papers. So the most cited papers of this wonderful year of Einstein is this paper on Brownian motion. And here, the motivation of Darwin is to prove that atoms and molecules exist. So fundamental. And in doing so, he set forth the basic diffusion equations and show how we can measure the movement. And this is the link here, because in a random walk, the mean location of the particle that is moving in random walk is in distance 0 from the origin. Okay, it will move at that side, and then it will move randomly to the other side, and so on and so forth. So the mean location is 0, but the displacement is increasing with time. And you are going to hear about the net square displacement, and this is the displacement, how far the particles depart from the origin in time, and Einstein showed that is proportional to the square of time, or the net squared displacement is linearly proportional to time. Okay? So this principle was brought forth by Einstein. He did not prove the number of atoms in mole substance. It was Perrin who got the Nobel Prize for that. But that was a very important contribution for Einstein that you will hear in these 5 days about random walk models and so on. This was the very beginning. Also Pearson in the same year wrote a paper and he put the name random walk. So these are the foundations of some of the principles that we discussed. And we face these great challenges. In 2005, Science asked some scientists, leading scientists, to put forward the major challenges, the most difficult and important questions that are still not resolved. One of these questions was the question of navigation of long-distance migrants. And I guess that scientists all over the world, if they are asked what is the main challenge in science, they will bring their own perspective and perspective of people that study movement, as far as I can testify, are these very general questions. So what determines the repertoire of movement modes used by individual population of species? Why we have this diversity? It's like thinking about species diversity and trying to understand that. So do the same exercise for the huge diversity of movement phenomena that we have. So what drives the evolution of these different movement phenomena, and what are the basic components that differ, in which movement differs between taxonomic groups or any other way of classification that we think. And we are not even close to answer these questions. And the incentive to go after these questions is got the general name Movement Ecology. And now it is a good time, excellent time for movement ecology to look and develop the study of movement of organisms in order to put the fundamental element to develop the general theory. So why it is now a very good time for movement ecology? First, because we have new technology. So now we can quantify movement in much higher detail and longer time than it was possible before. And this is changing every month, every week. We got new technologies that are pushing us forward. Something that now we conceived as regular, and we do it now on everyday basis, were not available even 2 years ago. And then there are new data analysis methods and tools that facilitate new insight into the mechanisms that underlie the movement patterns. And there are new frameworks to conceptual frameworks to set the stage for develop this general theory. So this part in the middle is actually the subject of this course, and so I'm not going to elaborate on this. You will hear a lot all these 5 days about the new tools, and we are here to learn and train you on this data analysis tools. The technology, I'll give you just not all the newest technology, but I give you just a short window to just illustrate these advances. And radio telemetry was invented about 60 years ago, and those who practice radio telemetry knows how difficult it is to do so. It takes lots of labor, lots of time. You end up the whole day with 10 people running like crazy in the desert, and you have 12 points that are not so accurate, and we are still limited with small animals to this kind of technology, but now we have much better tools. It's mostly that can also, we can also track much smaller organisms, and also is involved with this study of butterflies, in which with harmonic radar you can track for certain distances of few hundred meters the movement of very small organisms. Of course, GPS is a major player in this development, and there are some other tools that I'm going to illustrate them for this bird. Okay, this is a griffon vulture, and this is a study in our lab of Or, Ro'i, and Alex. And this is with collaboration with Wayne Gates and with Ada Tzofeh. So here you see the wing tag, and this is not very new. Birds have been tagged, and then they can be identified. This was actually the standard way of monitoring the whereabouts of these animals. Now we have RFID tag here in this wing tag. This is a project of Ro'i. Where's Ro'i? Is here? Yeah. And so now we can automatically record the presence, and because this is very reliable, we also know the absence of these birds in selected size, and it is much better, you know, even if in some location once a year you got observers in one location and they are looking for the animals and the tags. We have shown that the RFID system identifies many more individuals that the very skilled observer have identified in the same place, and now consider that we got position every second or every minute, and every day, so 7 days a week, and in a site, and so this is a major advance for presence absence data of animals that can carry RFID tags, and they are getting smaller and smaller. Here we have a GPS tag, and with GPS now we can get very accurate positions. We can play with the trade-off between sampling frequency and duration. And you will see this data later on. On the same tag, devices to measure acceleration. From acceleration, we can identify behavioral modes, energy expenditure. I will show that. And then we take also blood or feeders and extract DNA. And from the DNA, we can know sex, relatedness, maybe the source of these individuals, and also we can do some other chemical analysis of hormones related to stress and so on. So for these track individuals, we have plenty of information today. We now study the importance, the role of information centers in vulture colonies. And actually, we have an information boost in every one animals that we track. Just to give you an example of these tools, from the acceleration data, we can define in good distinction different behavioral modes. You see the signature of the acceleration in 3 dimensions is quite different between these different behaviors. And then we can tell the sex and the relatedness between individuals. And these 2 individuals here, a male and a female, are special because these are 2 vultures that did what we call long-range forays. And to know what long-range forays are, these are a summary of tracks of 43 or 44 individuals that we have. And they are mostly trapped either in the Judean desert or the Negev desert, and they move here in this area locally. Sometimes they go to the Golanites and back. Sometimes they go to Jordan and back. So these are, we consider, local movements. But some individuals did these long forays to Saudi Arabia, to Sinai, and down to this Asir mountain region close to Yemen border. And when we test this, we found out that it's 5 females and 1 male. And this 1 male is the brother of a female that was doing so. And another bird is this one that was known in the media as the spying vulture. And so this spying vulture is, you see our device, it was actually a female too. And then given that we have these strong bites to females who are doing these very long movements, whereas the female to male ratio is exactly half in the population and also in our tag individuals, we ask what drives these females to do these very long forays. And just to give you a short summary of the stage of research is that these are those individuals doing these long forays when they do this in the commuting flight. So they start here in the Negev, for example, and they do this commuting flight all the way, and then there's a foraging phase here, and then they come back in another commuting phase. So in a commuting phase, we have them here in black, and in dark gray, we have them in the foraging phase of this movement. And then we have them in this light gray when they, the same individuals are foraging in their home range. And then we have this other individual in the same time in the home range, and you see that during this forage they travel much more, not only in the commuting phase but also in the foraging phase. So every day they travel much more, and this is how much they eat. So how can you tell that a vulture was eating when it was in Saudi Arabia? And this is the strength of this tool. We have this acceleration data. And this was here in this place, you know, in Asir Mountains, very far from here, in November 8th. And in this place, we got this signature, and this is a signature typical of vultures eating on a carcass. So we know that this is a feeding event, and we can tell that they eat much less during these forays. So they travel more, they eat much less, and then they expend more energy. This is ODBA is a major derived from the total acceleration. The more the animal moves, it is expending more energy. So it's an indirect measure of energy expenditure, but logical, and it has been it has been tested in several cases. So why they do this? Why these females are doing these long-range forays, despite the fact that they travel more, they eat less, they expend more energy? There must be a strong driver, and this is something that now we can formulate the question very clearly because of these tools, but maybe we need some more tools in order to understand it and answer these questions. It could be that they are looking for males of higher quality than the Israeli vulture males. It could be that there's some social structure that is in scales that we did not consider so far. It could be some genetic basis of this population, or maybe it's a metapopulation, a very large-scale metapopulation system. So this is something that, in order just to formulate the questions, we needed all these advanced technologies and also discover this phenomena. Okay, so now I'm going to this conceptual framework, and our contribution to this framework was done by this team. And these are the people that were in this Movement Ecology group that convened for 1 year in the Institute for Advanced Studies. And that was a wonderful year. The Institute of Advanced Studies, that is the next building here, provides wonderful facilities for groups to do science. So they gave us all these opportunities, including trips and good time together. But they demand that we sit together quite a lot and think about the problem. And the problem is how we set the stage for development of a general theory for movement. And it was actually introduced to the general community, scientific community, before we started this year. This was a special feature in 2006 in Science, and there's a newspaper that by Candice Holden that she heard about this forthcoming project before it started, and she interviewed several people involved with this project, and now she said that a few researchers said it's time for some synthesis, and the perspectives were not very optimistic. This is why she called it inching, which means moving very, very slowly towards some goal. So we got this how you call that, promotion or this very nice display of this initiative in science. But what actually made this happen is this year that we work very closely together, and eventually I was invited to edit a special feature in PNAS, and the contribution in this special feature were half from the member of this group and half from people outside the group, like Otso. Contributed a wonderful paper to this collection, and and that was the introduction of movement ecology in 2008. And this is what I would like to emphasize, that this is a proposal. It's not the theory itself. It's a proposal how we can get to develop this proposal. So it aims at simulate the development and sharing of research tools. This is what we are going to do in these 5 days to promote the understanding of the causes, consequences, mechanism, and patterns of movement, and of course to set the stage for the development of a unifying theory. The idea is that if we can think mechanistically about different movement phenomena and understand them deeply in common language, okay, so we can hope that we will have the insights to develop this general theory. So how you do that? One of the first decisions was to focus on individuals. Okay, it is movement, for example, in plants was always considered a population level process. Very few people consider the movement of individual seeds, and that's true for many systems. But the movement is done by the individual, and that was one of the first decisions we make, to look what individual is doing, why it moves like that, and so on. And the second is to consider the scale and the phases of movement. So in ideal world, we have what is called the lifetime track of an individual. So we know every step in its lifetime, from birth to death. Okay? Imagine, and we are getting there. We now have much better technology to quantify this, not only in small, very small, tiny organism under microscope, but also in large, wide-ranging, free-ranging individuals. So now, this individual in this day was searching for food here, and then escaping a predator here, and then continue searching for food here. When you look in the finer detail, then you got the steps. And this hierarchy is, here is a lifetime track of a famous store called Princess. Almost lifetime tracks of about 300,000 kilometers. And these are the steps, and we can think about the metaphor of a whole genome sequencing, in which the lifetime track is equivalent to the complete genome, and these steps are equivalent to the nucleotide that makes this genome. Now, the revolution made by this, by our ability to get the whole genome and the basic components, is, everybody appreciated, one of the major contribution of science in the last centuries. And and then what we really need to know is, what is this functional region? Okay, this is supposed to be a gene or something functional. And in movement path, the equivalent is a movement phase. So here, there's a phase of searching, and now there's a change in phase. There's something very different. And this is one phase, and now there's another phase. Now, the way that we look at the movement path and identify these phases is what we call segmentation. Okay, and Luca is going to talk about that. And we want to segment the movement paths into functional units that are different, and we can, this is a major challenge in movement research. And then what we also want to do is to think, what are these very few basic components that are applied for every kind of movement. So here in different colors, you have all these kinds of different movements: migration, dispersal, foraging, and so on and so forth. And now they are given in different colors, so think about how colors are made of, there are 3 or 4 basic colors that with different combination they make these different varieties. So this is the metaphor that we want to understand what are the basic principles, the basic components of movements. And this is our proposal of these basic components. It's not the only one, it's not the best one, it's just one trial to do so. So we start with the internal phase of the individual. This is what the individual, the energy, the physiology, the mental state of the individuals, from which the most important feature is the motivation to move. So why this individual is moving now from here to there? Because it's hungry, because it's afraid, because of different things in its internal state. And then when there's a motivation to move, you need a capacity to move. This is the motion capacity. This is the machineries that, you know, the muscles and the skeletons that allow us to move, or the wings of birds or wings of seeds and so on. And then movement is not random. In many cases, it's not random in space and time. And then we need some machineries to perceive information from the environment and decide when and where to move. And these mechanisms are called navigation mechanism. And these all apply for the individuals, for one individual, and then all other things in the world. Other individuals, other species, physical factors are included in this one box that is called external factors. And when we put these pieces together, we got this graphical framework. Again, we start with the internal phase, the motivation to move, and then we need some capacity to move, and this by itself can generate the movement path that we see. And but as we say, it often goes in this way. So there is an ability to perceive the environment and make decision and kind of give instruction to the motion capacity where and when to move, and then you generate the movement path. And this path has feedback to the internal state. The animal now runs away from the zone of fear, and now it is free, so its internal state is now different because of the movement. And of course, it can also affect the external factors, and the external factors are everything. They affect, you know, other organisms, other members of the group, other species, competitor, predators, the physical structure of the environment, the wind, the temperature, and so on, and they affect each one of these components, of course. And so with that very simple framework, if we can have now a certain study system and say, okay, this is now C dispersal. And in C dispersal, we can identify and name the parameters that relate to each one of these components. Now we go to bird migration, and we can do it. Now we go to whales, or to albatross foraging, or to the movement of C. elegans in a petri dish. And in each one of these very different systems, we can tell, this is now we can identify the internal state, the motion capacity, navigation capacity, and the main external driver of this movement. And this is, the idea is that if we have this common language and common framework, then we can gather the insights that are needed to develop the general theory. And what I think was really interesting in this process, that happened one day, or even the same day, that we come up with this framework that took us 7 months. This is so simple, but and you've seen all these highly talented and famous people in the group, and we were thinking, what is the most simple way and it took us 7 months to get to this. But then when we look at that, we recognize that it maps the main paradigms of studying movement. And take, for example, biomechanics. Biomechanics is about motion capacity. You know, how horses are running and how cheetah is running and how seeds are flying in the air and so on. It's all about that. This huge field of research, which is excellent, which is insightful and everything, never, almost never, or even never looks at the navigation capacity on the external factors or the movement path. These components are completely ignored because this is a field of science devoted to the biomechanics. Now the same holds for the cognitive scientists, and here this is now these machineries to perceive the environment in the brain and so on are the main focus, and how the movement is generated by the motion mechanism and what is the movement path is not considered or seldom considered in this kind of studies. And then a third paradigm is the paradigm of what we call random. This is when you look only on the path and you try to understand everything from the path itself. So you think that everything is in the path and will tell you. So people that are doing that are coming mostly from physics. And we had a member in the Movement Ecology group, Yossi Klafter. So Yossi Klafter did some very important contribution. He defined the Levy walk and the Levy flight that you're going to hear about. And he was telling us all the time that what we need to do in order to develop movement ecology is to find the simplest possible track. And he said the simplest possible track that you can think of movement of organism is sea dispersal by wind. Because there's no nervous systems, there's no position of the environment, the abiotic factor is very simple, governed by very simple rules. So if you will be able to understand the movement of seeds by air, then this is like the hydrogen atom. You solve the structure of the hydrogen atom. You don't need to go to all other 100 and something elements. This is the main insight that you can get, and for me it was kind of frustrating because I invested so much on sea dispersal by wind before I started movement ecology, and I couldn't feel why is that enough to understand everything. So, but this is a way of thinking, okay? It's very different. Biologists think differently from people from physics. I think we can appreciate the importance of extracting as much information as possible from the result of all these processes of the movement path. But it's not sufficient. And the fourth paradigm is what we call optimality, and consider, for example, optimal foraging, optimal migration, optimal dispersal, well-developed theory, wonderful theory. All these subjects are movement phenomena. Migration is movement, foraging is movement, and dispersal is movement, and they almost never really go into the movement itself. But they ask questions like, under what condition the animal will get away from a patch, okay? You know these famous theories. And under what condition the animals will minimize the time or minimize the energy during migration. So how they do that, and how they navigate, and how and what is the movement path is not very is not part of this line of research. So what we think is that all these paradigms are not new, but what is new is the integration. The movement ecology is trying to get all these components and trying to make the study of movement legitimate. You don't need an excuse that movement is important for community structure or for population dynamics. It is legitimate to study movement per se, and this is a field of science that is opening this field to study the movement itself with all its varieties and so on. So I'm moving to the second part of my talk with, I actually two examples that I would like to give for these general principles from the study in my lab. So there was a long tradition of studying seed dispersal by wind, mostly, also by birds. We were engaged in study of ant movement as well, but most of our current research is about birds, bee eaters, vultures that you have seen, cranes, storks, stone curlews, jackdaws, barn owls, and also mammals. So from this work, and many of the people who are leading this project are here, I'm going to focus on two different systems. And of course, all these studies are wonderful, so I'm not selecting the best studies. But I want to select studies that will emphasize some of the points that I think are worth emphasizing. And I will start with an example that will not be discussed in this 5 days course. This is plant dispersal. And there are three main reasons that I'm doing that. First is that I think that the difficulties of applying the movement ecology framework to plants is important. We need to address these difficulties. You will see that it's actually not that difficult. Second is that I think it shows that we need to integrate between scales, and how we can do it, how we can integrate between scales. So in this case, I will move from a temporal scale of 20 minutes to hundreds of years. And also because in this, like Yossi Klafter said, it's a system where we have advanced the science to good understanding. So we are in good position in terms of the understanding of the mechanism. So now we can pursue even more difficult questions. And this framework is, of course, the seed is the individuals that we focus on, and the wind is the most important external factor. And there are all these adaptations to facilitate dispersal by wind. And imagine that you can track these seeds in the air. It's almost impossible for all species, but at least in simulations you can do it. So you can also have the movement path. So it is easy to quantify these three components, or at least with models. But what is the internal state of a seed? How can we tell that the seed has any motivation to move? And how it navigates? We're thinking about animals, so we can think about the nervous system and decision-making processes. And so how we put plants in this framework. I think it's quite easy if we think about this framework as in evolutionary terms. Okay, if we put it in an evolutionary perspective, then the motivation is set by the selection to find establishment sites. It's not during the lifetime of the seeds, but it's during many generations. And this selection actually is deriving all these adaptation, okay, including this adaptation, which is those traits that determine the timing of seed release in relation to dispersal vectors and establishment of opportunities. So there is a selection to find establishment opportunity in time and space, and it can be achieved by facilitating movement itself, and also by the synchronization or the timing of the movement. So if we think about this framework not during the behavioral within life perspective that we use for animals, but from longer multiple generation evolutionary perspective, then it's quite easy to put this context. And in this study that was part of the special feature in PNAS, we ask what determines different strategies of seed dispersal by wind in a tropical forest in Panama. And two species that we selected are Tabebuia rosea, which is one of the many, almost all species in this forest are dispersed in the dry season. It's between February and April or May, and you will see why. But this weird species, which is quite common, is called jacaranda, dispersed in the wet season. So we wanted to understand why actually jacaranda dispersed in the wet season. And so if we talk about the navigation capacity, so this is one aspect of the season of dispersal, and this is another aspect of finer resolution. And we found that the seeds of these species and many other species are dispersed mostly in relation to strong winds. That the dry season is typically of stronger winds. Whether the seed release in 20 minutes intervals is better explained by the sensible heat flux. And this is explained, this is the number of weeks in 2005, 6, and 7, and this is the peak dispersal of Tabebuia. This is the peak dispersal of jacaranda. And these are the different components of turbulence. This is a convective component, which is always quite low. Okay, it gets some peaks, but it's generally low. And this is the mechanical component, which is very strong in the dry season. As you can see here, much stronger in the dry season than in the wet season. So jacaranda timing is related to a period in which the convective features of turbulence have higher proportion compared to the overall. It's not dominating, but here it's much higher and has bigger impact in each of these September, October month. And this means, and another component of the difference between the species is the motion capacity, and part of it is the terminal velocity of the seeds, the rate of fall in still air. And Tabebuia is a normal 1 meter per second, more or less, terminal velocity of almost all seeds of wind dispersed trees. And jacaranda is very low. It's one of the lowest terminal velocity that you can ever quantify for most seeds, and that's not including poplars and so on that are very tiny seeds, but many trees which have very large seeds like this one, and so now we have a very large wing area which makes this possible. And now we go into this insight. So we need to understand the motivation, okay, so-called, of the seeds to disperse. And this is the selection to find establishment opportunities. And if they are successful, we can quantify the dispersal success. How successful was this selection? And we do that by comparing the dry and the wet season, or Tabebuia with jacaranda. And for example, with dispersal distance, it's higher than day one, so Tabebuia has a longer dispersal distance. So if dispersal distance is a good measure of dispersal success because they escape the enemies near the mother and so on, and they have higher probability to find establishment site, then the prevailing strategy of dispersing in the dry season is we can understand this, okay? So this is maybe a major force for most species to disperse in the dry season because they disperse farther away. But if we look for other measures of this motivation, there should be a selection for an individual that put the seeds in more sites, right? So we will have advantage over other individuals. And also, and here jacaranda wins. It's marginally, or it is small, but significant difference. And also, in terms of the mother plants, a better strategy is to put the offspring in different locations farther away from each other so they will not compete each other. So this is quantified by the distance to the nearest seed. And here jacaranda is also doing much better. So if these are the terms of dispersal success, then jacaranda is doing better, and we can understand this different strategy and why they reach more sites and they are farther away from each other despite the stronger winds. The reason is that the wind direction in the dry season is very narrow. So the winds are going from east to west, most of in the middle of the day. So seeds are getting farther, but they are only in this section. Whereas in the wet season, the winds are for different directions, and they are weaker, but they put some seeds here, there, and so on. So this is why jacaranda is actually taking advantage of the wind conditions, the special wind conditions of the wet season. And here we show by analysis of variants, these variants here, this is each species, jacaranda and tabebuya, with its own traits, and this is the proportion of seeds that got uplifted. It's a measure of dispersal distance, because uplifted seeds disperse farther. And then and here we just took the old traits of jacaranda and only changed the season. Okay, so now jacaranda is dispersing in the dry season, not in the wet season, but with all other traits the same. And you see that this change is not changing anything. Its performance in terms of uplifting percentage is the same. Okay? But what is critically important for jacaranda is this very strange trait of very low terminal velocity. Without this trait, it will not get the seed uplifting and disperse farther away. So this strategy is actually the timing to get the benefits of the adirectional winds, and the way to do it is strong selection for low terminal velocity, which has its own trade-offs with establishment and so on. So therefore, we can understand why only one species was selected to take this strategy. But what I'm trying to emphasize here are the two components of the motivation to move and the navigation capacity of plants. It was essential to understand that we can apply these terms to plants in order to do this analysis and understand these strategies of seed dispersal. And I'm continue, you know, this was based on models and empirical measurements every 20 minutes. And now I'm jumping to hundreds of years and asking the questions about plant response to climate changes. And these are some of the climate envelope models that predict that these plants in 100 years or less, they will shift the range towards the north, and you can see that under different scenarios of climate change, they are supposed to move 2 to 4 kilometers per year in order to track this change. So these are the shift between the centroid of the current distribution and the projected distribution. And this is true not only for plants but for many animal species. And And this is a new analysis published in Nature a few years ago, and this is the expected velocity of climate change, the surface temperature, how fast it's going to change when you look at the geographical gradients, and you see it is in the order of hundreds of meters in these forest ecosystems. A few hundreds of meters is maybe too much for plants to track, especially trees that need to grow. It takes 10, 20 years for a seed to become a reproductive tree. And the best way you can think about the movement of the population spread of plants is this framework that produced by Jim Clark, which is called invasion by extremes. So now we have the dispersal kernels, the probability of dispersal distance, and then the farthest individuals, not the 95%, but the farthest individual dispersed by this population is the one that is going to establish to a tree very far from the population, and then this one will establish a local population and will disperse from this point. And it has been shown that this is the fastest possible scenario for dispersal, but the problem was that it was based on dispersal kernels that were estimated from seed traps. That this is actually one of the few ways you can estimate dispersal kernels. And seed traps are, you can put seed traps 10 meters, 20 meters, 60 meters, 100 meters, really exceptional, but nothing in the scale of kilometers. It's impossible to do seed trap studies in scale of kilometers, and therefore what you quantify for limited area is not necessarily extrapolated well to larger scales. And also the parameters are quite synthetic. So we have, for example, the 2DT of Jim Clark. We have two parameters, and so it's one parameter is 1.5 and another is 2.8. What is the meaning of this? It has a meaning in the shape of the dispersal kernel, but no mechanistic meaning. So in order to tackle this problem, we use mechanistic models. Mechanistic models, you plug parameters that you can quantify and understand, like terminal velocity that we were talking before, and the height of release, and the wind speed, and so on. So these are different types of models that become more and more sophisticated and advanced and complicated in time to this large eddy simulation that Gil Borer was doing, that got us wonderful insight into the role of canopy heterogeneity. But this simulation takes about 2 months to simulate half a day of dispersal. It is impossible with this kind of very good insight into the mechanism of seed dispersal to even think about questions of population spread. That needs years, hundreds of years, thousands of years, and even more. And the solution came with Gabi Katul finding this analytical model that is mechanistic. As far as I know, until today, this is the only analytical model that is mechanistic. And so this is the dispersal kernel, and it has a shape and scale parameters like all other dispersal kernels, but these shape and scale parameters consist of parameters that we can understand and quantify, and these are the very simple parameters like mean tree height and terminal velocity, wind, and so on, turbulence. So we can quantify all this, and now we are in a good position to develop a mechanistic model in which the parameters of dispersal are included, the parameters of the demography, like the very important one, the maturation age, how long it takes for a seed to become a reproductive tree, and and with that, we can plug the projected changes into the model, okay, which is impossible with other kinds of dispersal models. For example, in this experiment in Duke Forest, in phase experiments, we this group, it's again Jim Clark from Duke, have shown that in plots where there's enrichment of CO2, okay, you got twofold increase in fecundity. So pine trees in these plots here, where they release a high concentration of CO2, are producing twice as much cones and seeds compared to trees just here in the control areas. Okay, so we can conclude from that the higher CO2 concentration in the future, trees will produce more seeds, and that's logical. Okay, they need a carbon to put, and they use extra carbon to produce more cones and more seeds. Not only they will use more seeds, and they will also reach maturation earlier. So they also use the extra carbon to develop faster, and they reach maturation 7% earlier. And another components that we can consider for the future is that in some areas wind is projected to increase, and some areas is projected to decrease. So this is a sensitivity analysis of the model parameters. From left to right, you got the most influential parameters and the least influential parameters here. So the most influential one is the maturation age. The earlier you reach maturation, the faster will be the spread, okay? Because this cuts down the length of the generation between successive jumps. And then the post-dispersal survival is a second one. And then when we look at this projection that I showed earlier, then we know that maturation age is going to be shorter, so trees in the future will spread faster. Okay, that's a prediction from this model and from this empirical data. We know nothing about post-dispersal survival. So we did sensitivity analysis in 6 orders of magnitudes, because we really know nothing about it. It is very low, but how low, we don't know. 1 in 1,000, 1 in 10,000, 1 in million. Not 1 in 10 or 1 in 100. It's not really likely that so many trees will survive in such high probability. C-terminal velocity is the third influential factor. The slower the seed is falling, then it has a higher chance to be dispersed farther away. So it has a negative influence, and we don't know if that will change with climate change. But we know that fecundity is going to be higher in the future. So they, again, are predicted to move faster. And then with the mean horizontal wind speed in areas where the wind speed will increase, they are supposed to spread faster and otherwise slower. So you see that in general, we can think that maybe there's a tendency to trees to spread faster in future climates. But when we do this sensitivity analysis, this is the 6th order of magnitudes that we vary the survival. And this is the condition in which seeds are dispersed. So here they are dispersed in any wind, and here only in strong winds. And this is the spread rate. So we got these hundreds of kilometers that we want to see in the future only in these very unrealistic cases in which the survival is extremely high and all seeds are dispersed in very strong winds. In all other scenarios, which are much more realistic, I would say that real life is somewhere here, we get a typical wind-dispersed tree that will spread only 10, 20 meters a year. And when we do that for different species, this was for a typical species. Now we do that for 12 different species. You can see that we got these values only for these 3 species that have different combinations of traits that facilitate a high spread. But for many others and many other scenarios, and this is very strong winds with very high survival. So again, we got this very unusual and unrealistic combination. So the main conclusion is that from the mechanistic perspective, we cannot anticipate that most trees will be able to follow these changes. And now I'm moving to the last component of the animals. And this is a project on food bats that Asaf Zohar was leading. Now Asaf Zohar is working in the National Protection Agency, and now David Shoham is leading this effort. And we started with food bats in order to understand the dispersal of seeds by food bats. So that was the beginning of this project about 7 years ago. But soon we understand that these creatures are moving in a fantastic way, so it became now the focal species, and then the fruits and the trees are the important external factors. But this switch is actually brought us to think that if we use the movement ecology framework and we talk about assisted movement, like passive dispersal by wind or by animals or by or migration or spread of pathogens by birds and so on, then we need to think about a nested framework in which the movement of one organism is nested within the movement of another organism. So we need to understand the components of these 2 systems in order to understand the whole process. So this is Asaf Tzotzo. He's taking out a bat from the net, and you see that these bats are quite nice. What I would like to emphasize in particular is the big eyes. Okay, most of people perceive bats as blinded or semi-blinded or almost blinded, but these bats have very good vision ability, and we put what was in that time the smallest GPS, and you see how small it is. It is a bit higher than the percentage that we usually do, and we did it before because mother bats are taking the babies with them, and they fly with them quite well. And you see here a mother bat with a baby here, and they can weigh about half of the weight of the mother. And in any case, we believe that our bats were doing okay. We got the tags and the bats okay for the time of experiment. So we put the tag. This is my daughter and Tzotzo. And this is the bat, and now we release the bat, and now from the folklore of this study, how you find this GPS, it's over there. It's not this one. This is the receiver and the antenna, so it's here. And now we get closer, and it's here, and now we get closer here, and now you see. So you really need some special skills like Tzotzos and Yoav and Ophir who were working on that, and to find this. And sometimes it's in these weird places in the caves, and you see Tzotzo is so happy to find. And so overall, they look like that. They are chewed by some rodents before we find them, but in most cases the data is okay. And the data is actually tremendous. So we did some radiotelemetry work, and we got with very hard work this kind of data. And for GPS, we actually or we, I was saying it's mostly Tsotso and Yoav and Ophir, were able to retrieve most of the tags. So we got about 80% of the data. And so now we are going to see what we get, and this is an example for what the new technology can bring us. So we are going to depart from this near the cave of where we did the study, and now we trap the bat, we put the GPS, we give it some food, and then we release it after 20 minutes. And what we see now is the track of this bat. It's the real data. There's no interpolation here. It's 1 hertz and a point every second. And it flies in about 35 kilometers per hour in about 140 meters above the ground. And you see that the path is really, really directional. It knows exactly where you want to go. Here in Kfar Daniel, he spent 20 minutes in this tree, 20 minutes in this one, 20 in this one. Now he takes, look how he flies above this border between the fields, and then takes a low elevation flight to this particular tree here, about 25 kilometers from the roost. And when you come to the next day, you will see the bat in this particular tree. And when you come the other day, and up to 4 months, David found that they even return to the same tree up to 4 months. It's not, we are not, you know, the quantification of this return is not strong because these GPS tracks work between 2 and 4 days. So between 2 and 4 days, we know that quite good, but over a longer time, it's more eclectic information, but we know that they do return for the same tree for a long time. What is special about this Morus tree here is unknown. There are many, many other trees around. Some of them are visited by other bats, some of them are not. But anyhow, what we found out is this amazing navigation capacity you have seen from the moment he got out of this of the roost. You know exactly where it flew, and this is the roost, and these are the beloved trees of all of these 32 bats. And you see that the foraging area is restricted, and you see these long sticks of the commuting flights from the cave to the beloved tree here, here, and so on. So these are the characteristics of movements that we are going to talk about in this course. So they fly in very straight path. This is the median speed. The displacement between the cave and the furthest point is about 13 kilometers, the median, and this is the mean altitude. So they do go to the same tree day by day, and then this project become from sea dispersal by bats to a question about the navigation of the bats, because we were astonished to see this behavior. So we did this typical translocation experiments of taking an animal from a location where we know there is a goal, and this was very good that they have this beloved tree, and this is the very strong destination all the time, or most of the time. And then we did a translocation, and if they rely on route following, they will not find them, and then they will get lost and exhibit this kind of path. If they rely on some directional based mechanism, they are supposed to fly in the same direction to the goal, and they will not reach the goal because of the translocation. And if they have what is called the true navigation capacity, they will know that they are here, and then they need to fly in this direction to the goal. So this is a very typical type of experiments. So we took them to the desert, where we believe that there's no bats in this area. This is the first experiment that Tsotso did. This is a place near Beersheba. It's in the northern part of the desert. Not many bat population, if any, in this particular area. And this is the area where they are. So half of the bats were released early at night without feeding. We expected they will fly to their beloved tree. So this is the path of the first bat, and this is the tree that he got. And the second day, you see this very straight line, very straight flight, exactly to the same tree from the cave. Now, the second bunch of bats were released late at night, and after feeding, the expectation is that they will fly to the roost, and that's what they did. And the same bat was foraging the next day here in this area. So this gave us some faith that we are in the right direction, and you see the different paths of this experiment. In general, 5 of the bats that were expected to reach the tree did so, and 4 of those that were expected to reach the cave did so. But there were exceptions, and look at that. Now the bat is released here near Beersheba, and this is real data. So immediately the bat flies in the right direction, really, within seconds. So the flight is not as straight, but it's more curvative, but it's northward. And this is a bat released late at night, but not late enough. So this is the cave, and now you see a decision-making, what we can speculate is a decision-making event. So here we make a decision not to fly to roost, but to this tree. This is, of course, the beloved tree of this bat. It's 15 kilometers from the decision point. And then it spends about 6 minutes here in this tree, probably not to feed, but to see who's around and so on. And then it flies back to the cave. So this is again showing you that how much insight we can get now that we can get the movement so well quantified. And then we ask, what is the mechanism that they use for navigation? Olfaction is an option. There are food bats. They smell the fruits, but it is unlikely that the odors of the particular tree can be perceived from 44 kilometers, but maybe the whole region has a special signature, and maybe the winds coming from the sea has special smell. Another option is magnetic field. In this study area, we have the largest anomaly in this area, and this is interesting. We are going to study these effects. And another option is that they use visual landmarks. And this was commented by pilots that heard about this study. Amit Dolev, if you know him. So Amit Dolev told us that actually from Giv'ot Goral near Beersheba, when we release them, you can see lots of lights. You can see junction. You can see many very clear visual landmarks that maybe they can see also from their home range. So we release them to a place which is probably unfamiliar, but they still can see familiar landscapes. And then Tzotzo did this line of sight analysis, and all these green spots can be seen both from the home range and from this release point. So it's not 100% true navigation because, huh? I don't remember the details. I think it's the typical height of, or even the maximum, or I don't, maybe 95% of the heights here, and here it's from 100 meters. But you see that there's a lot of overlap. So they can see familiar landmarks. And then we need to find a place where they cannot see that. And in Israel, we have this geological phenomena that is called erosion crater. It's a big hole in the ground. It's about 400 meters deep. And this is about 10 kilometers. This is about 4 kilometers. And when we release part of the bats from the bottom, they can see no visual landmark in the coast. there's no way to see anything but the walls of the crater. And this is Mount, how's that called? Avnun, Mount Avnun, and from here they can see. It's farther away. It's 84 kilometers from the roost. And this is, in short, the results. But released from the crater were lost. So they did long path within the crater. They were not moving in the right direction. Up to 40 kilometers they fly in the crater. Whereas bat released from the mountain just 4 kilometers from this location. They fly straight, you see that. This is the formal analysis of these populations. All bats are moving. This is the distance from the release point, and this is the cumulative straightness index. So they immediately start to fly in a very straight line, and it got very straight, after a few kilometers. But in the crater, you see that they turn left and right and so on, and only when they get out of the crater it returns to the straight flight home. So with that, we believe an important point was that eventually all of them left the crater in the north side. All of the bats that were lost in the crater, eventually they left to the north. So we perceive that as a signature that there's a secondary mechanism. So visual is, of course, shown here experimentally that it is. Huh? What's that? Yeah. I'm not in favor of this mechanism because they fly well in cloudy nights, and they return from Beersheba and from here in every night in equal performance. So visual is important, but there's a secondary mechanism that we don't yet understand that operates when the vision is prevented. And this is, as far as we know, the first time that we have evidence for true navigation or large-scale navigational map in a mammal. So I'm going to finish with this movement ecology. The state of the art is that the field is progressing very rapidly. This is the search of the specific term movement ecology, and you see that the number of papers and number of citation is increasing in last years. And this is the PNAS paper, and this is the state space models paper that also co-authored. And you see these are the two most cited papers, and in this course we are going to talk about state space modeling. And I just give you some examples that maybe we did not anticipate to see movement ecology studies of these kinds. This is from last week. Roy Plotnick published this paper in Paleobiology, it's a journal of fossils and so. Trace fossils are those that leave traces that tells something about the activity. And Roy is saying that this community needs to consider the movement ecology paradigm in order to understand the behavior, to link behavioral ecology and the study of fossils. Another quite interesting extension also published this week. Alex sent me this one a few days ago. So this is an attempt to unify movement ecology and biogeography. They show it for two African duck species, including this Egyptian goose, and you can see it, so you can read this paper, and they say that we need to understand these different components of internal state, navigation capacity, movement capacity in order to bridge the gap between understanding of mechanism of movement and the distribution of species. And they give a nice example for these species. And these are two other papers from this week. This one is also not very expected. The movements of hunters in the in the jungles of Ecuador by these people. So there's analysis of net square displacement, bias random bridges, and resource utilization functions of these hunters. And then there's another paper from a few days ago that used this red eye vireo in order to understand, and translocation experiment in order to understand. This is more mainstream work as like the others are. You know, this field is moving in many ways, and it's good, I think, because it showed the broad perspective. So I will end with this announcement that we are hopefully going to establish a new journal, part of the Biomed Central series. And with Luca, who's going to be your teacher, one of your teacher this week. And so that's my perception of the field and the basic principles. And I think I would like to thank you for your attention and to wish you all exciting learning and success. Thank you.
Great.
Questions? Yeah.
If you look at the seed dispersal, maybe you can also look at it the movement of the tree, not the seed.
Of the tree itself.
Yeah.
So there are some trees that move.
No, the seed moves via the seed. So.
Yeah. So you mean the growth of the tree?
The population of the tree.
No, it's actually the tree itself moving its genes, kind of.
Growth, you mean branches, and so there are trees that move. Really, there are trees that walk.
By dispersing the seed, you can look at it kind of as if he is moving.
Yeah. So the tree is moving when it is young, and we call this young tree a seed. Yeah, of course. But there are actually trees that move, that walk. You know, they have this kind of roots, and it has been shown in these canopy gaps that within years, trees with, how is this called? There's some system of roots that penetrate above the ground, and they move towards the light. So in 10 years, you can see a tree moving a few meters towards the gap. Yeah.
It's not what I meant.
Yeah, I know, but it's I agree. It's seeds are small trees,
No,
potential
the
small
tree
trees.
itself, by spreading its seeds, it's kind of moving.
Yeah, in multiple generation perspective. Yeah, I agree. Yeah.
About your paper in Ecology Letters in 2011, you based your modeling on the analytical solution that you presented before, but this solution is clearly Gaussian, right?
No, it's inverse Gaussian.
Inverse.
It's fat-tailed kernel. Yeah.
So you've answered my first question.
Yeah. That's the wild distribution. Wild and inverse Gaussian are the same distribution.
Okay. And the second question, about the bats, at least the two first routes that you showed had a little bias towards the sea at the beginning of the movement, and then they corrected back to the tree or the roost. Can you see any such trend for other of the bats? And this can also.
I don't remember. We
sound.
actually. David, you remember that? I don't think. No, maybe. Maybe. We haven't looked at that. Maybe they're shifted to see the electric towers of Ashkelon, maybe. I haven't looked at that. Yeah.
In the belt experiment, the second one with the Machtesh Gadol, did you try to see the points that could be seen like Tzotzo did in the first phase of how many visual points could be seen from the Machtesh Gadol and from the normal path?
No, but in general, the flight within the crater was very low.
I mean the one on Har of Non. Did they have
I know.
any point that they could see from both sides?
Yeah. So it should be done, but in general, the flight was very low, and we were expecting that they will do this kind of climbing up and then see, and then, but they do not. They usually fly very low, like they fly on the way back, which is 10 meters, in the order of 10 meters, and that's what they fly almost throughout this getting lost part. But formally, we did not test any.
So is it possible that maybe the ones that you assume that they go from the cave to the feeding point, they have from some times that they do some other circles, and so they get to know more than you were expecting. And so for them from Har of Nun, they can see some places those unusual forays that you see.
Yeah. Maybe they gather information about. It's possible that they gather information about visual landmarks. It's very unlikely, I think, because as I said, the flight is low. But maybe they during these movements they gather information about other cues. So maybe it needs a scale of 5 or to 10 kilometers to sense for example, the gradient in odors. So if they do that back and forth, then they can sense it, or maybe even the magnetic. The inclination is very structures, south to north, but maybe they cannot feel the magnetic field in few hundred meters. But they can feel it in several kilometers. So these are possible mechanisms that David is going to test with manipulation and so on. So it's not a closed story. It's just the beginning. Yeah.
So do you think that city lights could be part of the visual landmarks?
What's that?
Do you think city
City.
lights can be part of the visual
Yeah.
landmarks they use? But electricity only exists a few hundred years, and those beds are much
Yeah.
older than that.
But then this is most notable visual cues today. But in ancient times, I think the notable visual cues were mountains that they can be seen in the night, and so the topographic structure can be seen at night, and big animals, big elements probably were always there. Maybe fire, I don't know. Yeah.
Nobody displaced them hundreds of years ago.
Yeah.
Do you see a difference in moonlit night and non-moonlit night?
No,
No difference.
no different. All right. Okay, what is the plan now?